So we are pleased to welcome Daniel Garcia, researcher in Paléobiology at the National Centre for Research of Human Evolution of Burgos in Spain. Daniel was awarded in 21 under the third transnational access call of Synthesis Plus for a project to visit anthropology collection with X-ray tomography scanning, in particular on Neanderthal and Cro-Magnon ribcage, if I'm not mistaken.
Yeah.
Yeah, great. This project aimed to better understand the evolutionary anatomy of the genus Homo from the Pleistocene to the present day by providing new information of the oxygen requirements and the behavioral transition of past population. Thus, Daniel was hosted for a week in December 21 by Dominique Grimaud-Hervé at the Musée de l'Homme and by Marta Bellato of the Asterix tomography platform. Daniel will kindly give a presentation of the progress of his work. So it's ROBUSTA project, the use of microcomputed tomography to delve into the evolutionary anatomy of the human ribcage. This presentation will last 40 minutes and then we will have 20 minutes for questions. Daniel, are you ready to start your presentation?
Yes, I am. Okay. So let me share. Well, first of all, thank you everybody for coming. And I'm going to start sharing my screen so you can see the presentation. Okay. Okay. Jonathan, can you please confirm if you're watching my screen?
Yes, it's perfect.
Okay. Okay, perfect.
And there is the square bottom of the screen. Yeah.
Okay. Okay, perfect.
Yes, great.
Okay, thank you very much. So again, thank you all for coming, and I'm gonna tell you briefly the project that we are ongoing, which is called ROBUSTA project, which is the acronym for ribs robusticity analysis, the use of a microcomputed tomography for exploration of the evolutionary anatomy of the human rib cage. So first of all, I'm gonna give you the index of the presentation. It will be 3 different parts. I will try to be brief in the 3 of them, but I think that is important to talk about the importance of the rib cage for human biology and for human evolution. Briefly talk about previous works that we have developed using 3D geometric morphometrics in the Neanderthal thorax, and then once you know the first steps, talk about the ROBUSTA project, the ribs robusticity analysis. So first of all, talking about the importance of the rib cage for human biology and human evolution, the rib cage, as you all probably know, but you know, sometimes the rib cage is not as well known and well studied as it should be. So the rib cage is a multi-articular skeletal complex which is anatomically integrated. So here we can see from Bastir 2008 and like a systems model definition in which the thoracic skeleton is in the axial skeleton, and this is linked to the cervical and lumbar skeleton, of course, in the upper part and in the lower part, but it is also linked to the skull, specifically to the face, because of the integration of the respiration on the nasal cavity with the rib cage, of course. Now we will see a little bit more of this. So we said that the rib cage is integrated with the fascial skeleton because, of course, the nasal aperture is linked to the oxygen consumption. All the oxygen that gets into the body through the nasal cavity is somehow used in the rib cage. So differences in the size and shape of the nasal aperture, of course, would be related to the lungs and to the rib cage, and differences in the nasal cavity would cause differences in the rib cage, hypothetically. Therefore, since the rib cage is obviously linked to the oxygen consumption, it's going to be linked to different paleophysiological aspects, such as the total lung capacity, the forced respiratory volume, and even the basal metabolic rate, because the energy that we expend is obviously linked to the oxygen that we can take into our body. So in here you see some images of a paper we published several years ago, in which different rib cages in Neanderthals in red and in humans in blue are probably linked also to differences in the craniofacial skeleton, such as the ones you see in Neanderthals in red and in humans in blue. When you superpose the rib cages on Neanderthals and humans, you see differences specifically in the lower part, in which is where the diaphragm is linked. But we will see more of this a little later. I just wanted to emphasize that the rib cage is obviously linked to these facts. Also, the rib cage is important because different rib cage morphology is going to be linked to different morphological variation in body shape. Like narrower rib cages are going to be linked with narrower pelvises, and wider rib cages are going to be linked with wider pelvises, therefore proposing different evolutionary models also. In here you can see, for example, one model for the evolution of the body shape proposed by Carretero and collaborators in 2004, in which you can see different body shapes in different hominin species. So again, this square that you can see here, the central square, is going to represent the rib cage. So the rib cage in all of these models are going to be important because of the body shape of the individual. Okay, once we have seen the obvious importance for the rib cage for human biology, I wanted to briefly summarize the most important works that we've done in this regard. First of all, I wanted to also say that the rib cage has been understudied compared to other anatomical regions, and this is caused by several problems. First of all, there are several problems that we have solved when studying the rib cage anatomy. So first of all, the methodological limitations of the 3D shape of the ribs, the methymeric nature of the rib cage, which causes that sometimes we just found a couple of individual ribs from the entire rib cage, and also the fragility of the ribs. So therefore, even though the rib is something like that, we usually find ribs like that, okay? Oh, sorry. Very broken and taphonomically distorted, etc. And this is one of the best cases. Sometimes, usually we find even worse cases. And how do we measure the ribs? Okay, so traditionally, ribs were measured using arcs, chords, angles, etc. But these traditional measurements, even though they produce a lot of information, they also have some limitations. So first of all, the 3D morphology of the rib is very complex, as you can see here. So when you use chords, angles, and arcs, there's some information that you don't get. So there's a need for 3D quantification. So what we did for that was developing a protocol of landmarks and sliding semi-landmarks that I developed with Marcus last year in my dissertation for quantifying the rib cage and also the individual ribs and individual vertebra. As you can see here, we followed a protocol of landmarks and sliding semi-landmarks for doing that. So using this landmark protocol that I mentioned, I did my dissertation specifically on the rib cage of Neanderthals. We know there's a correlation between the cranial and facial shape and the body shape in Neanderthals and modern humans. And as you see here in this skeleton that you can see on the right hand side, the Neanderthal body morphology and the ribcage morphology is very different from the one from Homo sapiens. But what is the reason for that differences in body form and in thorax shape, specifically between Neanderthals and modern humans? You can see here a reconstruction done by a paleo artist between showing the differences between the Neanderthal torso, the Neanderthal thorax and pelvis, and the modern human thorax and pelvis. And we will see this in more detail in the next slides. But you see that the Neanderthal body shape is much wider than in the case of modern humans, caused by a wide pelvis and a wide rib cage. But why the Neanderthal morphology is like that? The Neanderthal thorax and torso morphology is like that. There's the first hypothesis called the ecogeographical hypothesis, in which Neanderthals evolved in a colder climate compared to modern humans. So because of the ecogeographical rules of Allen and Bergman, the Neanderthal morphology was like stockier than in modern humans, like wider and shorter. This is the first ecogeographical hypothesis. But there's also a bioenergetic hypothesis, which is the second one, proposed among others by Churchill in 2006, but this has been proposed by many other researchers before. So this researcher proposed that the body mass, the basal metabolic rate of Neanderthals was much larger than in modern humans, caused partially by largely consuming energy tissues, such as the brain, but also the muscles. Neanderthal muscle mass was hypothetically larger than in modern humans, and these two tissues, the brain and the muscles, consumes a lot of energy. So this is why hypothetically the ribcage should be larger, wider, and more efficient than in modern humans. Also, there's another proposal by Ben Dorr and colleagues in 2016 that talks about the liver, the size of the liver as an important factor also. And this could also need for a lot of energy. So hypothetically, the ribcage will produce energy for this large liver, following this hypothesis by Ben Dorr and colleagues. Anyway, probably the Neanderthals needed a larger oxygen intake compared to modern humans because of the brain size, muscle size, liver size, whatever the reason. Neanderthals needed larger oxygen intake compared to modern humans. So the first Neanderthal that we studied in my dissertation was the Guevara 2 Neanderthal of 60,000 years ago from Mount Carmel in Israel. Here you can see the burial of this Neanderthal, but when you see the individual ribs, you see that it's very well preserved. The rib cage of this individual is very, very well preserved, as well as the pelvis and other skeletal remains. But this individual, even though is the best preserved Neanderthal in the fossil record, it has some problems. First of all, the original reconstruction of the ribs was partially wrong. Here you can see in blue the original seventh rib of this individual, and in red and in green you can see the correct reconstruction of the seventh rib, which was done by Asier Gomez Olivencia in 2009, and we reconstructed in 3D. So in here, in the right hand side, you can see the ribs from 1 to 10 in the x-axis, and in the y-axis you can see the size of the ribs. The green line is the original Neanderthal size, and in the blue line, you can see the correctly reconstructed ribs. So you can see that in this part, the Guevara rib cage is actually larger than initially proposed. So when this issue is solved, there's a second issue, which is Guevara 2 is the best preserved Neanderthal, but potentially it has a costal anomaly or pathology. So in here you can see the 3D model that there's some called endocostal ossification in the ribs, which could be potentially pathological. So before studying this individual, it was important to see that it was not pathological. So studying these 3 ribs in a comparative sample and with a lot of medical literature, we finally see that probably it was something called Meckel's costal anomaly, which is a non-pathological costal anomaly. It was not a fracture, it was not a pseudoarthrosis, it was probably a non-pathological anomaly called Meckel's costal anomaly. So once we knew that it was not pathological, we could start studying the Guevara-2 analysis, the Guevara-2 ribs. Also, we combine this information from Guevara with the information from El Cidrón. El Cidrón is a Neanderthal site in the north of Spain, in Asturias, close to Gijón, for example, and it's a 49,000 years old fossil site. Specifically, we started studying the thoracic vertebra of these Neanderthals, and you can see here level thoracic 6 and thoracic 7, and you see Neanderthals in green and modern humans in blue. You see that the transverse process orientation, among other features, is very different between them. In the blue line represents the modern humans, and the green line represents Neanderthals. And you see that the orientation of the transverse process is much more dorsally oriented in Neanderthals compared to modern humans. But we are going to see what does this mean. It is important to follow what we call a holistic approach, because the sum of the, not studying only individual ribs or individual vertebra, because the sum of the parts can produce emergent characteristics in the whole, in the entire thing. So individual parts can produce complex 3D morphologies or complex anatomies. So what happened when we link the ribs with the vertebra? Again, this is work that we did on the ribs from the El Cidrón site. This is a 5th rib and a 11th rib reconstructed, and when we articulated the vertebra with the ribs, we start seeing differences between Neanderthals and humans. So specifically, we see here we see in blue color Neanderthals and in red color modern humans, and we see that the Neanderthal ribcage was probably wider and maybe deeper, as you can see in here, but specifically wider in this central lower part. This allows us also to, in collaboration with Asier Gomez Olivencia, to reconstruct the Neanderthal thorax morphology of the Guevara-2 Neanderthal, confirming that, again, the Neanderthal ribcage was deeper, as you can see here in lateral view, but it was also wider, as you can see here in frontal view. In a second step, we followed an ontogenetic analysis in order to study different individuals to see if the difference that we saw in adults were also differences from early ontogeny. So we worked with the Roch de Marsal, the Lemoustier 2 individual, the Mesmaiskaya 1, and the Derrier 1. This was part of my first postdoctoral experience at the PASEA institution at the University of Bordeaux. And thanks to this postdoc, we could study the early development of the Neanderthal ribcage, confirming that there were differences in between Neanderthals and modern humans ribcage from birth, basically. In here, you can see a principal component analysis, observing that the trajectories of Neanderthals and modern humans were parallel. But when we compare the average, which is the blue line of modern humans, and the orange lines of Neanderthals, you see that there are enough differences between them. Neanderthals are located more to the negative scores of PC2 in this case, and this is caused by an expansion of the lower ribcage and a deeper also ribcage. So our conclusions from this study was that there were differences between Homo sapiens and Neanderthals at birth, at least in the ribcage, probably also in other parts such as the face, the skull, the nasal aperture, but this work is specifically on the thorax, because the thorax of Neanderthal newborns was wider, deeper, and shorter compared to modern humans. Okay. But once we knew that the Neanderthal ribcage was different than modern humans, probably caused by differences in ecogeography or bioenergetics, we wanted to know if those differences in the ribs could have a biomechanical significance. And this is where the ROBUSTA project comes, in order to study the ribs robusticity analysis. Okay. So first of all, I wanted to say that the bone robusticity is directly tied to biomechanical loading and stress that the bone undergoes, and therefore changes in robusticity are usually understood as the result of the evolution of human habitual activity and mobility. Here we can see two examples of the work from Chris Raff. In this part, in the left hand side, we can see the femoral anteroposterior strength and the tibial anteroposterior strength. And we can see that the strength of the femur and tibia has decreasing from the upper Paleolithic to nowadays. And this can be observed in the femur. Okay, you see that the femur of upper Paleolithic humans is stronger and the tibia of upper Paleolithic humans is stronger. And it can be observed both in males and females. The blue line is male and the red line is female. So you see that the long bones strength has been decreasing from the upper Paleolithic to nowadays. This is observed in the robusticity, but it's observed also in the shape of the bones. You can see here also from the work from Raff and Larsen in 2014 that there are differences in the cross-section of the femur between pre-agricultural and agricultural humans. So the study of human long bones, for example, the femur or the tibia, has proposed that the increase in the sedentary lifestyle during the Pleistocene-Holocene transition was the major cause of a decrease in human skeletal robusticity. But we wanted to know if this is also, okay, some authors have observed this in the long bones, in the femur and tibia, but we wanted to know if we can observe also this in the ribs, because we saw that the upper Paleolithic, like the Paleolithic ribcage of Neanderthals is different, but we wanted to know if maybe we can also observe something like that in the ribcage. So changes in physical activity and mobility due to the increase in sedentary lifestyle should be obviously reflected in the oxygen needs and respiratory dynamics of the individuals, thus impacting the ribs, mechanical stress, and robustness. In here again, we can see the picture from Raff and Larsen in 2014, where they observed differences in the cross-section, and in a very preliminary study, we also observed differences in the cross-section of the ribs between Neanderthals and modern humans. Specifically, you see here a principal component 2 analysis quantifying the cross-section of the rib. We have a thinner cross-section in the negative values and more rounder cross-section in the positive values. And here you see that males and females of modern humans present thinner rib cages like this, but we studied other Neanderthals such as Shanidar, Kevarachu, Taboon, La Chapelle, La Ferracie, Krapina, El Cidrón, but also we studied some fossil Homo sapiens and even a Homo antecessor. So when we studied this, the cross-section morphology, we saw that Neanderthals overall have a rounder cross-sections compared to modern humans, but fossil modern humans, which are these that we see here, are somehow like between Neanderthals and modern humans. So probably their cross-section, the ribs cross-section, were somewhere in between a thin and a rounder cross-section. But this is very, very preliminary, as I said. So in order to study this cross-sectional anatomy and the density and the robustness of the ribs, we are studying microcomputed tomography. With the microcomputed tomography, you can study the same that studied Raff and Larsen, for example, in 2014. You can study the same in the ribs without any damage or without destroying the ribs, which is sometimes in order to study the cross-section, you have to carry out classical histological techniques, and you need to actually cut the rib. But we didn't want to cut the rib. We wanted to study the internal anatomy and the cross-section anatomy without any damage. So here you can see several dicom slices from a first rib of modern human, and you can see here that you can study the internal anatomy of the rib, the thickness, the robustness, and the cross-sectional anatomy in different parts of the rib without any damage. And then, how can you quantify or how can you study the rib density or the rib robustness? So this is a pilot study that we've done with a software called Dragonfly. In this software, you can differentiate between the cortical and trabecular bone, and you can quantify the area. For example, this is the mid shaft of the rib, quantified through this protocol. In blue color, you can see the cortical bone, and in orange color, you can see the trabecular bone. And this is something that we aim to do here in this project in order to study the changes in robustness of the rib and changes in the density of the rib. But we can also do like an even more complex study or more complete study, studying the bone density in the whole rib, not only in the mid shaft of the rib, but in the entire rib. This is a protocol from Profico and collaborators from a couple of years ago called MorphoMap R package, and this protocol was developed for the femur. You can see in red color in this femur the thicker or the more dense parts, and in blue colors the less dense parts. And this potentially can be also applied to the ribs in order to study if there are different densities of the ribs between Neanderthals and modern humans, between different populations of modern humans, etc. Specifically here in this rib, you see again in red color the more dense parts, and in blue color the less dense parts. The red parts corresponds to the muscle attachments of the scalenus, serratus, etc. But also to the articulation. In here you can see the rib head is also red. So this is the articulation of the rib with the vertebra. So this is something that we aim to do in the ribs of modern humans and Neanderthals to observe if the ribs from this species was also different from ours. So our project has two main objectives. The first one is to compare changes in rib robusticity in our species with the increase of sedentarism from Neolithic to nowadays, or even we can study this from the Upper Paleolithic to nowadays. And the second objective is compare changes in rib robusticity between Pleistocene human species such as Homo erectus, Homo antecessor, Neanderthals, and even fossil Homo sapiens. So this is where my work at the Musée de l'Homme had an importance, because I worked there getting micro-CT images from the ribs, from the Neanderthals, from La Ferracie 1 and 2, male and female, as well as La Sapelocenes. And I also got micro-CT scans from Abri-Pachot and Cro-Magnon, which are also important in order to study Upper Paleolithic humans compared to Upper Paleolithic Neanderthals, for example. But I have to say that this is still ongoing work. So scanning and analyzing is not a simple process. So it has a long protocol, and it's still ongoing. So hopefully in the near future, I will be able to tell you more about our results. Finally, I just wanted to acknowledge all the people that helped me during my stay in Paris, like Dominique Grimaud-Herbet, Antoine Valceau, Véronique Aurélie from the Musée de l'Homme also, and Marta Bellato from Asterix, and Jonathan Vettery also from Synthesis, for all the help and the support and the work from my stay in Paris. So that's all. As you saw, I just wanted to focus on the preliminary results that we got, and also what are supposed to be the next step of this project. And hopefully soon I will be able to tell you more about it. So it was like half an hour, something like that. So I hope I was not very very long. So thank you very much, all of you, for your attention, and I'll be happy to reply to any question that you could have.
Yes, many thanks, Daniel. You was very welcome to Paris, and it was a pleasure to welcome you here. Are there any questions?
I have no question, but I just want to congratulate Daniel for all this interesting work, because ribs are very difficult to study because they are very often fragmented and and very badly preserved. So it is a challenge. So
Thank you.
so
thank
thanks.
you,
Thanks for us. Thanks for us to do it. About the ecological condition that you speak about to explain the different shape of thoracical cage. You know that Neanderthals lives on cold climate, but also temperate climate. So how did you explain that? Because it can be an adaptation to cold climate, and after he stay like that. So
yeah, of course, I agree with you. And the eco-geographical hypothesis is just one hypothesis, but in my opinion, the larger energy expenditure and the larger oxygen intake explains much better the differences. Because, for example, in this work from Stephen Churchill in 2006, he found that Neanderthals living in hot, mild, and cold climates, in the three cases, they have a larger energy expenditure and probably oxygen intake. Because, for example, I don't remember well, but I think that other, like Amud, for example, he didn't live in a cold climate, if I remember well. And even Neanderthals living in hot or mild climates, they even had a larger energy expenditure, and probably they needed more oxygen in their bodies, so they needed the large ribcage. So in my opinion, the hypothesis about the energy expenditure is better than the eco-geographic hypothesis. So maybe for some Neanderthals, the eco-geographic hypothesis was right, but not for all the Neanderthals. I don't know if you agree, Dominique, but
yes, it could be an explanation. And so for you, the Neanderthals from Europe comes from Levant?
That's a good question.
Because all these remarks that you speak about comes from Kebara and other Levantine Neanderthals. So,
Also, it's important to test if, for example, Neanderthals from Levant, like Kebara and Shanidar, if they are exactly the same compared to the Neanderthals from Europe, like the Alcidron, La Ferracie, La Chapelle. Actually, in this work, Stephen Churchill also proposed that it could be differences between Levantine Neanderthals and European Neanderthals, and this could be also important to observe in the ribcage, if we can observe the exact same differences between Levantine and European Neanderthals. This is an important point, and it could be tested in our study if the internal anatomy or density of the ribs is the same in Levantine Neanderthals and European Neanderthals. This is an important point, yeah.
And you do the relation also with the long bones of inferior membre, comment on dit la jambe, quoi. And so perhaps you have also to compare with Homo erectus, because the cortical tissue on Homo erectus is very thick, and there is perhaps you can find an explanation if you compare both species. But there is no ribs with Homo erectus. Is there?
Yeah, it's the only the Nariokotome boy, but the problem is that it's a subadult. It's a juvenile adolescent.
It's a child. It's only 8 years old.
Yeah, exactly. So we can study the Nariokotome boy, but we won't know if the differences that we are finding are because it was an 8 years old, or it is because it's a Homo erectus. Because we see that there are differences in evolution in the density of the ribs, but there are also differences in ontogeny, in the density. So younger ribs, like ribs from childs and juveniles, are more dense than the ribs of adults. So if we study the Nariocotomy boy, it will be difficult to know if the more density of the ribs is caused because it was a Homo erectus or because it was a subadult. So yeah, it is, and also for the case of another possibility for studying lower Pleistocene hominids would be studying the Homo antecessor, but in Homo antecessor you don't have individuals because it's everything very mixed up. So you cannot have information from the rib and femur from the same individual. So yeah, this is a pity. It is complicated.
And it is also more complicated because Nariocotomy, you say that it is an Homo erectus, but it is an African Homo erectus.
Yeah,
so,
of course.
So African Homo erectus and Asian Homo erectus are very, very different.
Yeah.
So for me, Nariocotomy is not an Homo erectus, it is an Homo ergaster.
Yeah, exactly. Yeah,
so,
you're right.
Okay.
Yeah.
Okay.
You're right.
So
but
I finished, Jonathan, if somebody wants to speak.
Okay, thank you.
Thank you. Anyway, thank you very much, Daniel, for all your work and your tenacity. And
thank you.
You're welcome.
Thanks a lot.
Okay, someone else want to take the floor or not? Okay, I think it's the hand. Daniel, do you want to add something or?
No, I just wanted to say thank you for all your help, and I really enjoyed my stay in Paris. I always enjoy when I stay in Paris, not only because the Tour Eiffel, also because of the fossils. And I always enjoy a lot when I'm there, and at the collection, it's a great collection, and I'm always very happy to be there. So hopefully we will have interesting results soon to show you. So, and I hopefully I can go back to Paris at some point. So I'll be very happy. Thank you very much.
Okay, you will be welcome too. Once again, thank you, and wish you all the best for the next step.
Thank you.
And that's it.
Okay.
Thanks to all to be here, and have a nice day.
Okay, you too. Thank you very much. Merci beaucoup.
Merci à toi.
Ok, au revoir.
Au revoir.
Au revoir. À bientôt. Au revoir.
À bientôt.